second part of lecture 2, and in this part I'll talk about improvement of strain. Okay, so after you isolated your desired organism or desired microorganisms, you can improve it. Okay, so make it better for using to the using in the industrial as an industrial strain. Okay, so and we can do this by different way. For example, by selecting natural variants with more potential ability. Okay, and or we can induce mutation into that isolates and select a better mutants, or we can use we can use recombinant DNA technology, okay, to make a better recombinants. Okay, so now I'll talk in more details about these ways to improve the strain. First, natural variants. Usually the natural isolates, okay, their yield capability is usually low. That means they produce commercially important products in low concentration. Okay, usually very low concentration. However, we can increase the production or yield by optimizing their growth condition. Okay, by adjusting the temperature, pH, agitation, oxygen level, carbon dioxide level. Okay, or by providing the best nutrients, okay, optimum nutrients composition. Okay, in this way we can increase the production of the strain. Okay, and then while we do this thing, while we optimize the condition in that, and during doing so we can choose the best or superior quality natural variants. Okay, however, this kind of natural variants have a drawback. So what kind of drawback? When these cells divide, these are microorganisms, so they will definitely continuously divide. So during division, heterogeneous culture, that means heterogeneous microorganisms can be produced, and if heterogeneous culture is produced, the yield may get degenerated. Okay, so that's why actually is very important. Whenever you deal with natural variants, you have to maintain this culture very carefully so that they don't so that heterogeneous population are not produced. Okay, you have to maintain them very carefully, and regularly you have to check for quality control purpose. To check the quality of the strain. Okay, so this was about natural variants. Then induced mutants. So we can induce mutation into a microorganism by using various mutagens, like we can use physical mutagens, X-ray, UV, radiation, or we can use chemical mutations. So by using these mutagens, we can induce mutation into our isolate, and then we have to assess different mutants. Okay, and finally have to, and through our assessment, have to find the best strain. Okay, so and what this best strain means? These strains will have the characteristics of, will have our desired characteristics, such as they which produce more metabolites. Okay, which has increased permeability. Okay, and which do not produce feedback inhibitors or repressors, or which do not recognize inhibitors or repressors. Okay, and finally, uh, by finally recombinants. Okay, so as I said, we can use different techniques, such as recombinant DNA technologies, recombinant DNA techniques, to make recombinants. So what recombinant means? When we introduce a foreign gene or genes into a cell, it is called recombinant cell. Okay, so like for example, we want to improve some trait, or introduce some new trait or characteristics into our microorganism, then we can just transfer a foreign gene, which have a foreign gene, and introduce that foreign gene into the cell. Okay, and make them recombinant, and we can do this kind of things, there are different techniques for to do this. Like for example, recombinant DNA techniques, protoplast fusion techniques, etc. Okay. Anyway, whenever we talk about strain improvement, the the first thing that come into our head is we want to increase the yield, but it's not always the right thing, right? So sometimes we may get our desired uh, strain through isolation, which produce enough yield. Okay, the production, which produce our desired metabolites in enough concentration. But still, we may need to change it some of the characteristics, or we may need to introduce some characteristics, okay, some traits other than yield production into that microbes. Okay, so what kind of improvement are those, or what kind of traits, such as for example stable strains. Okay, we got the strain, it produced enough metabolites. Okay, but it's not that stable, so we need to improve it so that it become stable. Okay, or we can make them resistant to some certain infection. Okay, and non-foaming strain. Okay, because during the fermentation, foaming is a problem. Okay, so if the strain produce too much foam, it actually it affects the efficiency of the fermentation process. So if we can modify the strain in a way that it produces less foam, okay, the fermentation process will be more efficient. Okay, and also like strains which are resistant to components of the medium, morphologically favorable strain. Okay, so suppose your strain is a very sticky type. Okay, so it's actually difficult to ferment do the fermentation with sticky cells because they tend to stick with each other. Okay, so we can by using the modification improvement methods, if you can make it less sticky, if you can make your cell less sticky, so it will be more easier to do the fermentation with these cells. Okay, and strains which are tolerant of low oxygen tension. Okay, and elimination of undesirable products from a production stream. Strains producing new fermentation products. Okay, so this was about the improvement of strains. In the next part, I'll talk about development of inocula for fermentation process. Thank you.